So the first movie I wanted to show you was about the model system, and this is a talk using Zebrafish as the model system. We have used this particular system to understand what happens very early during embryonic development. And the reason Zebrafish is chosen is very elaborately shown in this movie, which is basically the first 10 hours of the life of zebrafish condensed into 30 seconds. And as you can see, when I play this movie, you will see that the early development is in form of rapid cell proliferation. The cells start dividing and forms a bulk of cells, and now they are overcrowded, so they start migrating. That's how one can also study cell migration, and this process is called epiboly. And the cells start moving, and then they invaginate. This is the early gastrulation, and this fundamental process is extremely essential to form the 3 germ layers. As you all know, we arise from these 3 germ layers. Different cell types are arising out of formation of this type of cellular folding event, which is the early gastrulation, which is essential because you need to have a sandwich. So there is an ectoderm, there is a mesoderm, and there is endoderm. And these 3 cell layers give rise to various cell types that we know in the adult cells, in the adult organism. So as you can see, within the first few hours, you can see the formation of head-like structure, the tail, and the somites, which will give rise to other organs later on. So this beautiful structure that you saw just in a flash in front of you is a very orchestrated phenomenon of the early gastrulation. And we probed into this because we wanted to understand, as the cells are dividing and they're acquiring progressively the signals to specialize into different types of cells, how are these achieved at the level of gene regulatory networks that keep on changing? So we probed this by looking at these properties of these cells. So classical experiments in the 50s and 60s using classical embryology, a large number of developmental biologists came up with a fate map of the embryos, as you see here in a very nicely colorful map. Just like the map of our globe, you can see that there are different countries here, different types of tissues that can arise from this part of the embryo, okay? So obviously at this point, which is not even completely an epiboly state, you can see that the cells have not specialized. They are almost identical to each other. Nevertheless, they have something inherent in them which can give rise to a different specialized cell type in the adult. So you can actually remove the cells from here and transplant them elsewhere, and they will be true to the cell fate that is already ascribed to them. So how does it happen before they are specialized? How does it get ingrained into these cells that what they're going to be? So it's like mapping of their fate, and that's why these are called the cell fate maps. So this is that early epiboly stage. After gastrulation, you can see that you still see a very distinct boundary for each of these. So these cell fate maps were very nice. However, the molecular underpinnings of this entire process, what are the molecular cues that these cells contain, was not clear. So to understand this, we took the approach of single cell biology. So just like the data you saw earlier this morning. So we took these embryos and separated the cells and looked at the expression of individual cell types, and they clustered into these 8 clusters after UMAP clustering. And you can see that one of these clusters, they are in different colors, but this one, which is circled, actually shows intermixing of number of different types of gene markers. Now these markers are for various types of different germ layers. For example, we can see the SOX proteins, guscoid, which is for axial mesoderm, endoderm. So all of these markers are expressed in this particular cell type, which means this particular ball of cells, this bunch of cells, has not been specialized enough to be able to separate it into individual clusters, as you see here. So what does this mean? So now if you overlay this information back to the cell fate model that I showed you earlier, at the molecular level, the information looks a bit fuzzy, not as distinct, not as spatially distinct as you saw in the first fate map that I showed you. So if you plot the molecular markers that are known to specify endoderm, mesoderm, or ectoderm, they are not as distinctly occupying different cell types as it were supposed to be when these cells will become the adult cells. So you can see here, this map is very fuzzy. So this dichotomy is something that really appealed to us, and we wanted to probe this further, and that's what today's talk is about. How does, at a molecular level, different types of gene expression patterns can specify the cell fate determination in a developing embryo. So the 3 specific questions that we asked in this particular study are shown here. We wanted to understand the linear specific chromatin landscape at the onset of gastrulation. We wanted to investigate the role of chromatin organization, the chromatin organized proteins, what is the role in vertebrate embryogenesis, and finally to understand the interplay between the organizers and the adhesion proteins by looking at the cell-cell adhesion, because that plays a significant role during this entire process of early gastrulation in the early events of vertebrate gastrulation. So I will not talk about the last one. Today I'm going to focus mostly on the second one, especially because of the shortage of time. I'm sure all of you have heard of Waddington's hypothesis. Those you know epigenetics, Waddington was the first one who actually could articulate very nicely what do we expect as a result of the epigenetic kind of modifications. So he proposed a landscape, and what you see here is a very sophisticated version of the Waddington landscape. What is shown here is like, this is like a foosball table. I'm sure all of you have played the game. You have the bumpers and flippers which flip-flop the balls across, and here the bumpers or flippers are endogenous versus exogenous factors which will kind of dictate the fate of the cells. So they can modify the GRNs, the transcription factors, the pluripotency factors, all of them are involved as part of the switches that is like a ping pong. So they actually move the fates, the activity levels of different genes and transcription factors to eventually give rise to a differentiated type of cell from an ES-like cell which enters somewhere here, okay? So it goes through all of this modifications, gets flipped and flopped, and as a result of whatever that happens, all the different types of epigenetic modifications that it will receive ultimately dictates the fate of the cell. So that is what is shown by this model very nicely, and there are different versions of this. So in zebrafish early development, what is known is summarized very nicely in this cartoon. As you go from the early embryonic development through epiboly and finally the fish-like organism, in the first 11 hours of development, there is a transition between early use of maternal genes which are deposited in the egg cell towards the zygotic genes. So the maternal genes, which are large number of RNA and protein molecules which are part of the egg, get utilized, and finally they decline rapidly after a few hours of development, which is 3 to 4 hours in zebrafish. So that transition is called zygotic genome activation, or MZT, maternal zygotic transition, which means there is a transition from utilization of maternal information to the zygotic information. This is also a very dramatic event in the life of an organism, because what is happening is the system inside the body is learning now to use the information encoded by the zygotic genome as opposed to what is donated by the mother, okay, as a part of proteins and RNAs that are given as gift for the early developmental effects. So this happens through a cascade of events, which is again very nicely mapped. It includes the pluripotency factors and some microRNAs, and most notably a microRNA called MIR430, which selectively degrades the maternal transcripts, and that's essential for early wave of zygotic transcription. So this is all very nicely known. However, what is also known is that during this entire transition of MZT or ZGA, there are large number of epigenetic events as summarized here. You will see that there is increasing DNA methylation. So this is the line that you should see, pre-ZGA and post-ZGA. You see that post-ZGA, the entire epigenomic landscape is completely transformed. You have all types of histone modifications, DNA modifications, and most notably, for today's talk, the increase in the chromatin accessibility that you see here. Chromatin is compacted here. Most of the regions are not accessible because those genes are not necessary to be activated. And then these genes will be opened up. They are amenable to various types of epigenetic events, the Waddington effects that you saw. And finally, that opening of chromatin leads to a variety of epigenetic modifications, which activates all the zygotic genes. So a number of single cell experiments were done on using the entire embryonic developmental model of zebrafish. However, there are certain caveats and certain limitations of the single cell experiments that people have used, most notably because you miss certain specialized type of cells, and secondly, the throughput, the depth that you need for analyzing some of the gene events is just not enough. So to circumvent this problem, what we did was we used a genetic tool where different cell fate markers, different germline markers were marked with fluorescent markers as shown here. And at different stages of development as shown here, shield is the earlier stage and bud is the later stage of gastrulation. You can see that at these two stages, we use these fluorescently marked genes, which are the reporter genes, to sort the cells, which are sometimes quite rare, less than 1% of the population, and collected. So we enriched the cells specifically using these fluorescent markers, sorted them using FACS, and then performed bulk transcriptomics on those sorted cells. So this required huge number of embryos because the total populations were really low, but we could then get large number of reads because these are enriched cell populations, and then perform this bulk transcriptomics to understand what are the changes, molecular changes. Remember the fuzzy image that I showed you, we wanted to pinpoint exactly, can we discover some new markers of cell fate determination? And indeed, we did discover some new markers. So most of the unpublished data is actually in the first part of my talk, and later on I'll show you some of the published information. We are working on this. Again, for the sake of time, I will not go through the details of each one of this, but nevertheless, when we looked at what changes at the level of chromatin proteins, we looked at various stages, different germ layers, what are the chromatin protein changes in the chromatin proteins, and two set of proteins, SATB family proteins, SATB1 and SATB2, which are chromatin organizer proteins. We found that it is SATB2 which is selectively enriched in the precaudal plate as opposed to SATB1, which is a related protein. We got particularly elated with this particular data because SATB2 is known to be involved in vertebrate gastrulation. So there is data from Xenopus which shows that SATB2, so this is an in situ hybridization, which shows that SATB2 is enriched at the dorsal organizer. This is the point at which the invagination happens during gastrulation, and that seems to be a critical juncture in early development of vertebrates. So we were quite excited about this particular finding and focused on SATB2, how it plays a role during this early event in zygotic genome activation. So if you look at the expression of SATB2 itself at various stages of zebrafish development, you can see that it shows a bimodal pattern, a small peak pre-ZGA, and there's a very big peak post-ZGA. These are two different stages. So you can see that this is the early stage and this is the organogenic phase where the somites and other organs are going to be formed. So we postulated that probably SATB2 might play two different roles at these two junctures, so pre-ZGA as well as post-GGA. So we tried to understand this by going deeper into both these phases. So first thing was to try to reduce or increase the activity levels of SATB2. So we took two approaches initially. First is morpholinose, which will downregulate SATB2 protein expression by affecting the RNA expression and the overexpression of SATB2. In both cases, again for this I'm not showing the movies, but you can nicely see that there is an embryonic developmental arrest at 80% epiboly if you knock down SATB2 using morpholinose. If you overexpress also, you get pretty much similar type of arrest much earlier, and there's a thickening at the dorsal. So which means that SATB2 is very critical during early gastrulation. So then what we did was we again, so this is a movie which shows what happens during the convergent extension. That's another very important phenomenon that happens early development in zebrafish, where the cells that have been invaginated from the blastopore, they migrate towards the anterior side to form the future brain, the nervous system. And that process now you'll see from the side of the developing embryos, and it is marked using two fluorescent markers. The nuclei are marked with H2B histone, which is fluorescent in blue color, and the precaudal plate mesoderm marker is goosecoid, which is marked with GFP, that's green. So if you see now what happens in the morphant, you'll see the complete zebrafish embryo, and the blue are the nuclei, individual cells, okay? They are moving around, and you're seeing now from the top, as opposed to the side view that you saw initially in the black and white movie, and you'll see the green cells emerging from top. So they have actually invaginated and moved across on top, and now they're moving ahead in a group of cells, which is the precaudal plate, and this is very important for formation of the embryonic early brain. And what you see here is that in the SATB2 knockdown, there is a severe defect as opposed to the control. So I will not show you the second movie because, again, because of shortage of time. The snapshots of both the movies are shown here, wild type versus the control. In the wild type, you can see that at various stages, the precaudal plate is formed very nicely. And if you take a section of this embryo, you can see that the germ layers are distinct from each other, very separate, okay, as if they are individual layers. In case of SATB2 knockdown, you can see that the plate is not as distinct as this. The cells are actually dispersed from each other. And most notably, if you look here, the two germ layers are no longer distinctly separated from each other. The cells are as if they are merging, interdigitating between each other. So there is a severe cell adhesion defect, which I mentioned, but I will not go into the cellular defect. We are going again into the molecular aspects of this. So we then generated a CRISPR-based knockout, and generating the homozygous took a lot of time because of the maternal effects, which is very complicated in case of zebrafish. So we had to adopt multiple strategies, and finally we succeeded in getting the knockout. And very interestingly, what it recapitulated is a defect that was known in humans as the cleft palate phenotype. Okay, there's a cleft palate in humans because of SATB2 mutants. Similarly, you see that in mouse also, and exactly the same defect was captured in the SATB2 knockout animals. You can see this is a micro CT image. Wild type is 3 times the size, smaller skeletal structures, and most notably, if you see the protrusion of the jaw and the cleft here, which recapitulates the phenotype in the humans and the mouse model, suggesting that now we can use these fish to understand what are the defects that give rise to the molecular changes. So when we looked at the molecular changes of gene expression, we performed another experiment. At these 3 stages, we took single embryos, okay, early gastrulation, mid gastrulation, and post gastrulation during organogenic phase, and we used tri-reagent. I'm sure all of you know that tri-reagent is used for typically isolation of RNA molecules, but it can be used for protein and DNA too. So we actually used tri for the purpose for which it was meant to be, to partition these 3 phases, and used this to understand, firstly, the organic phase as DNA, used it for genotyping because we don't know the genotype of the embryo, and then used that to isolate RNA from the aqueous phase and perform the bulk RNA-seq, and showed that there are a large number of changes in the 14 somatid stage, which is the organogenic phase. If you look at the differentiated, the genes which are differentially expressed, they are all part of the neural crest development, skeletal morphogenesis as well as neural crest. And indeed, this was quite revealing for us because neural crest are the early progenitors for formation of the lower jaw. And if you do not have the neural crest formation, there is some defect that will lead to abnormal lower jaw formation. The cleft palate is part of that. And we are quite happy to see that indeed the gene expression studies showed that that is indeed the case. Most notably, when we look at the overexpressed, the genes that are regulated by SATB2, it shows that SATB2 can positively regulate the neurogenesis and neural crest specific genes. And the NC2 patterns also showed the expressions of these genes very true to what is expected. So I'll just summarize this. When we looked at the occupancy after ChIP-seq for SATB2 across these phenotypes, we saw that SATB2 occupies all of these loci, SOX10, SOX9, etc., which are the markers of neural crest. Indeed, at very early stage, 80% epiboly, even before, so this is the 14 somatid stage that I talked about. So SATB2 seems to bookmark all of these sites even before they are determined to be a certain type of organ or certain specialized cell type. So this could be a molecular mechanism by which the kind of effects that I talked about early on could happen by bookmarking those sites even before this cell specialization occurs. So that's one indicator. And this last data I want to show you is about the difference between peripheral versus neural crest, to show very specifically that whatever defect at the neural crest we have observed is indeed the one that gives rise to the front part of the body, anterior structures. We did an experiment using mouse embryos. So in the early embryos, you can see that E9.5, where the 3 lobes of the brain are not specialized. You just take the entire head and perform the experiment using that. whereas in E13.5, you could dissect the lobes. We could take telencephalon, which is a positive control here, and then we perform ChIP-seq analysis, and you can see that at all these stages, head, trunk, and the dorsal telencephalon acting as a positive control, we see that in the cranial neural crest, which is in the head, SATB2 occupies these regions, but not in the trunk. Okay, and the same thing is true here. So indicating that it is the cranial neural crest and not the peripheral neural crest that is where SATB2 is responsible for dictating the gene expression profiles, and that's why the phenotype that we see, the phenotypic defect, is restricted to the anterior part and especially the lower jaw, which is a property of the vertebrates. Okay, so we have worked on the evolutionary aspects of this also, which I will not talk about today, but formation of the lower jaw was a very important evolutionary advantage and invention that has given rise to the vertebrates. So I will not go into this. We have done chromatin accessibility. I'll just summarize. The chromatin landscape undergoes massive remodeling during sulfate lineage specification, and the zygotic SATB2 potentiates early neurogenesis and neural crest specification by employing multiple mechanisms. So I hope I have convinced you. I didn't get time to talk about the accessibility data, but how do I go to the last slide? Acknowledge, yeah. So these are the people who did the work. Most of the work actually is part of PhD thesis of one talented graduate student, Saurabh, who is now moved on for postdoctoral work, and assisted by other people in the lab, the collaborators, as well as the funding agencies. Thank you.